Hi, welcome to the Aquatic Ecology Lab at Oak Ridge National Lab. My name is Terry Matthews, and I lead a group called Biodiversity and Ecosystem Health in the Environmental Sciences Division here. We're a diverse group of scientists with different expertise and skills, but we all focus on understanding the impacts of different energy strategies on the environment. And because we know that energy and water are so strongly linked, it takes energy to deliver and produce clean water for us to drink, and it also takes water to produce energy oftentimes. A lot of us focus on the impacts of energy on aquatic ecosystems. We're particularly focused on biodiversity, understanding the impacts of energy on biodiversity. Biodiversity is really just a term to describe the wide variety of life on Earth. Biodiversity is important. It's considered to be an indication of ecosystem health. We rely on biodiversity for things like clean drinking water or nutritious food, or even for disease control. So understanding how biodiversity changes with different energy strategies is really important. There are a lot of ways to study biodiversity, so we use a lot of tools in my group, from the molecular level all the way to the ecosystem level. Sometimes we need to study the environment and we need to know how many of a species are present, or we might need to know the health of the organisms that are present. And in that case, we will sample using traditional methods, so things that we've all seen. So fishing using fishing rod or nets. We sometimes use a technique called electrofishing, which just releases electricity into the water to stun fish temporarily. This just makes it easy for us to net the fish up and catch them so that we can measure them and look at which species and count which species are present. In addition, sometimes we want to look at things on a broader scale. For example, we might want to look at a forested area over time, or to look at submerged aquatic vegetation in a stream. We can use things like drones, either drones or satellites, to look at an entire watershed over time, to look at vegetation. My background is actually in oceanography, and I study how chemicals behave in the environment and how chemicals can affect which species can live in an environment over time. And these chemicals can be things like nutrients, like nitrate or phosphate, the things that you fertilize your plants with, but they can also be contaminants like mercury. And I'm particularly fascinated not only in how chemicals can control which species can live in a given ecosystem, but how the biodiversity or the species that live in an ecosystem can actually impact which chemicals and how the chemicals behave in the ecosystem. Mercury is one of the only metals that's known to biomagnify. And this just means that it becomes more and more concentrated as it moves up the food chain to fish. So our biggest dose of mercury comes from eating contaminated fish. The longer a fish lives, the more steps in the food chain there are, the higher the concentration of mercury in that fish. So even though you might have a healthy ecosystem, which is a diverse ecosystem, that means that you have more steps in the food chain. So in a healthy ecosystem, you could actually have fish that have high concentrations of mercury, which might not have been obvious if you just thought about it. I lead a long-term biological monitoring program, and we are sampling for biodiversity and water quality at the same sites over time. So at the same sites, we have entomologists that are collecting invertebrate samples to look at which species are there and how many are there. And we're particularly focused on looking for sensitive species that are sensitive to water quality, because we know that if they're there, then that water quality is good. We have ichthyologists that are doing the same thing, collecting fish samples to look at which species are there and are there sensitive species there. And then we are collecting water samples to look at the water quality, so to look at contaminant concentrations, nutrient concentrations. And we take some of the samples back, and we have toxicologists that actually look at how that water affects the survival and reproduction of both aquatic invertebrates and fish. What we found is that the water quality at our monitored sites has improved significantly over the 35 years that we've been monitoring. Not only has the water quality improved, but the biodiversity has improved significantly. So this is really great news. We're also developing technologies and strategies to improve the water quality. For example, we're working with material scientists and geochemists to develop things called sorbents that can absorb mercury and other contaminants from contaminated waters. And we do this in what's called artificial mesocosms, or artificial streams, where we actually bring in natural stream water from contaminated streams and then test out technologies in a flowing system. Because contaminants like mercury actually behave very differently in flowing water than in still water. So we're developing things like sorbents that can remove mercury so it cannot biomagnify. It's not available for uptake by fish. And one of the other things that we're doing is we are looking at the possibility of using native species of freshwater mussels. Mussels are really interesting animals. They are filter feeders, which means that they feed by pumping water over their gills to remove plankton and other particles for food. And these species are among the species across the world that are most in danger of extinction. Because they feed by filtering a lot of water, they're extremely sensitive to water quality. So we're working with Tennessee Wildlife Resource Agency to actually seed our local streams with mussels. These mussels are considered ecosystem engineers, which means that because they are filtering water, they're actually improving water quality over time, and they can significantly alter the ecosystem. So we're hoping that not only can we use the mussels potentially as a natural filter to remove mercury from the water column so it's not available for uptake by fish, but also we're restoring the ecosystem back to the way it was before there were impacts on the ecosystem.
Hi, my name is Louise Stevenson. I'm an environmental toxicologist at Oak Ridge National Lab, and today I'm going to talk to you all about the research that we do in the toxicology lab, or I'm going to call it the Tox Lab. I knew I wanted to be a scientist. I thought I wanted to be a marine biologist, but I ended up working on a research project with a professor in college looking at the effect of phytoestrogens on Siamese fighting fish. Phytoestrogens are plant estrogens, so plant hormones. All organisms have hormones, and they're actually very structurally similar across a lot of organisms. These plant-based estrogens serve a role in the plants, but when they accumulate at high concentrations downstream of pulp mills or places like that, they can actually pose an environmental effect on natural systems. So we looked at the effect of these plant-based estrogens on Siamese fighting fish, which are betta fish, which are fish that I imagine a lot of y'all have had at some point in your lives. You can buy them at any pet store. These Siamese fighting fish are really beautiful fish. The males have large fins and they flare. So they'll very aggressively flare. There are opercular fins, those are the fins by their face, or the fins on their backs. And that's a sign of aggression. And so if they see a conspecific or another betta fish, they'll think, oh, I need to show this betta fish who's boss. They flare, flare, flare. And you can actually put a mirror up to a container with a fish in it, and that fish will flare at what it thinks is another betta fish, literally until death. And so what we did is we looked at the effect of phytoestrogens on that behavior, and we found that with exposure to phytoestrogens, males actually became much less aggressive. And eventually, through future work, that lab was able to identify that decreased aggression was related to changes in the neurotransmitters in the fish's brains. But this research really led to my passion for environmental toxicology. I gave up wanting to be a marine biologist and decided that environmental toxicology was really what I was passionate about, because the problem of just one little fish in one little beaker in one little lab in one little college actually has applications to the whole natural world. So when I talk about environmental toxicology, I'm talking about the impact of various chemicals on natural systems. So these are usually chemicals that are introduced anthropogenically or by humans. You can think about these chemicals as everything from personal care products, so be some Tylenol or Advil that you take when you have a headache, or a prescription that you're prescribed by a doctor, vitamins, to things that you use around your house. So cleaning products, either for cleaning surfaces or thinking about doing your laundry, all the way to large-scale chemicals that we use for various applications. So applications like water sanitation, energy extraction, things like that. So when we talk about environmental toxicology, we're trying to get at the ecological risk that these various chemicals can have on our natural environment. So that's how I became interested in this field, and so I went on to get my PhD at the University of California, Santa Barbara. There I studied the effect of nanomaterials on freshwater systems. So when I say freshwater, I'm thinking about lakes and streams, water that's less salty than the ocean. Nanomaterials are very, very, very small materials. Nano is a prefix like micro or milli. It means 10 to the minus ninth. So these are super small things. This is on the scale of DNA. DNA is about 2.5 nanometers. These are very, very small particles that are engineered for various applications, and we were interested in looking to see if they had any sort of toxic effect on natural systems, and if that toxicity was different from what we call their bulk forms. So one of the particles that I looked at was nanosilver, and so silver is present in its ionic form, is the toxicity of nanosilver, very, very small silver, different from dissolved silver, bulk silver, things like that? So those are the types of questions that I study as an environmental toxicologist. Here at ORNL in the Tox Lab, which is where we are now, we do a lot of experiments relating to either regulatory testing, so permit-based tests that I'll talk about in a second, or more investigatory focused tests. So looking at the effects of novel compounds, specific chemicals, things that we're particularly interested in. So the regulatory based work that we do is using EPA or Environmental Protection Agency standardized methods. So these are methods that are very, very well standardized. They're very well defined. So that means theoretically, results that I get in my lab here in Tennessee are the same as results anyone would get in Switzerland, as Zimbabwe, as anywhere else, because we're using the same methods. And so they are very standardized, including standardization of what organisms. When we think about the effect of various chemicals on the environment, how do we actually estimate the effect? How do we estimate ecological risk? So one of the ways that we do that is we look at the effect of these chemicals on model organisms. So we look at the effect on various fish, or worms, or in my case, mostly plankton or zooplankton. And so we look at effects of various compounds on fish and zooplankton in this lab, and we use what are called model organisms. So the word model organism here means both. It's a model in the sense that this organism is being used as an example of all the biological diversity that is related to it. It's also a model organism in the sense that this is an organism that's studied really widely in the lab for a lot of various reasons. Biologists, evolutionary biologists, ecologists, a lot of different studies focus on these organisms. And so we know a lot about them and how they act in the lab and also how they act in nature. So the model organisms that we use are mostly fathead minnows, that's a fish, and then Daphnia. So our model organisms, we look at the effect of various compounds on the survival of these organisms, and then also what are called sublethal effects. So a lethal effect of a potential contaminant is exactly what it sounds like. Does it decrease the survival of that organism? A sublethal effect is something, in effect, that is less than lethal, sublethal. So these are effects on things like growth, reproduction. We could look at behavioral effects. There's a whole range of endpoints we could look at when we're trying to estimate the toxicity of a compound. For these standardized tests, we mostly look at growth for fish and reproduction for Daphnia. When we're doing a regulatory-based test, if any sponsor has a permit in order to discharge water into a natural system, so discharge effluent, then we take a sample of that effluent, and we want to make sure that it would not have an impact on the environmental system into which it's introduced. So let's say a wastewater treatment plant releases a bunch of effluent after it's been treated into a creek. We would take a sample of that effluent, we would test it in the lab, we would expose our model organisms in the lab to that effluent, and we would see if it impacts survival, growth, reproduction, and then be able to test, okay, does this effluent pose an ecological risk to the community? The more investigatory tests that we do here at ORNL are in collaboration with other scientists here. So we work with material scientists to develop new chemicals. So if a material scientist is developing a novel lubricant or something like that for an application that's going to go into the water, let's say, that material scientist wants to make sure that that lubricant is not going to have an effect on any aquatic ecosystems if it spills, if it gets released during normal use. And so that material scientist will develop that compound, give us a sample, we'll test the toxicity, compare it to what's commercially available, is it more or less toxic? And then iteratively, we've worked with scientists in the past, material scientists and chemists, to develop a more green chemical, so a chemical that has less of an impact on ecological communities. Also, in terms of investigatory studies, we're doing a lot of work trying to look at effects across biological scales. Looking at the effect of a contaminant on the individual level, does it decrease growth, reproduction, survival? We can also look at impacts on the suborganismal level or the molecular level of gene expression, changes in the concentrations of proteins or other things that are important for basic function of any organism. We can also look at effects on whole populations and communities, but these levels are obviously all connected. So as an ecologist and as a biologist, we're all really interested in looking at how can we extrapolate these effects across these levels. So one of the model organisms that we use in the Tox Lab that I was talking about previously are called Daphnia. So these are crustaceans. So they're crustaceans like crabs and lobsters, but much, much, much smaller. At maximum, they get about 4 millimeters in length, so very small. And these are really important prey items for fish, and they're also very ecologically important. So these are what we call filter feeders. So they filter water through their bodies in order to get food. They basically just throw food at their face in order to eat it. And so they interact with a lot of the water. So these are model organisms for the reasons that I was explaining earlier, but also because they serve as kind of a canary in the coal mine of aquatic toxicology. So canary in the coal mine, the idea for that was for coal workers, if the canary got sick or died, they had an idea that there was something in the air that they were being exposed to that was bad. Similarly for Daphnia, because they are filter feeders, we know that they're going to interact with a lot of the water, and so we use them as a model organism to get an idea of, okay, if this contaminant is in the water, is it going to have an effect on this very sensitive species? So Daphnia are what are called zooplankton. If you go into any freshwater body and scoop up a handful of water, you're likely to find them. Fish eat them, other invertebrates, those are organisms without a backbone, eat them as well. So they're used widely in environmental toxicology and evolutionary research and other similar research projects. The reason that we love them also for environmental toxicology is because they can actually reproduce asexually. So they can reproduce clonally without a male or any sexual recombination. So they reproduce clones. They produce literally clones of themselves. They lay eggs on their backs. The female Daphnia lay eggs on their backs into the brood pouch. Those eggs sit there for about 2 days. After about 2 days, they turn into embryos, so tiny little Daphnia with eyes. After 3 days of development, the little offspring, what we call neonates, those are babies, in the brood pouch of the mama Daphnia start jostle until they basically force the mom to molt. So they break what's called the carapace. So remember, these are crustaceans. So if you think about lobsters and crabs, how they molt and have a carapace or an external shell, that's the same idea of what we're talking about with these Daphnia. So the mama Daphnia molts, the offspring swim out and go on their merry way. So that's fascinating ecologically, but it's also really useful for environmental toxicology because it means that we can hold a single Daphnia, a single daphnid in a cup and look at the effect of reproduction through time. We don't need to introduce a male daphnid. The mama daphnid can do it on her own. And we can also have multiple cups of the female of these Daphnia and look at replication through time. So for a standard test, we'll have 10 cups at every treatment. So if we're looking at the effect of a novel chemical, we'll expose 10 Daphnia to the same chemical, and we'll get an idea of a sublethal impact on reproduction, and also the variability of that response. So maybe 1 cup reproduction is decreased by 75%, but in another cup, reproduction isn't affected at all. Then we have an idea that maybe we need to look further into this compound to get a better idea of its mode of action or how toxic it is. So that's one of the reasons why Daphnia are so widely used in environmental toxicology and are such a wonderful model organism.
I'm Christine Moody. I'm a molecular ecologist here at Oak Ridge National Lab in the Biodiversity and Ecosystem Health Group. I focus on using non-invasive molecular tools to understand the impacts that we are having on our environment, specifically how hydropower affects our fish communities and other aquatic species and their ecology. So eDNA is environmental DNA that organisms put off into the environment. It's through ordinary processes. Right now, you and I are producing eDNA into the air, and we can go out into the environment and collect this eDNA using different sampling methods. So we can sample water, soil, air, even honey, to look at what organisms are living around those areas and how many individuals of those organisms there are. We then bring those samples back to the lab and we process them in a way in which we can make more of that DNA. So we amplify it so that we then can use sequencing technology to look at what those samples are telling us. So our work combines laboratory work, molecular bench work, and field work. And so the lab that I'm in right now is our big fish laboratory. And in here we do mesocosm experiments, tank experiments, where we can put different fish, different number of individuals, and pull out water samples from them and do very controlled experiments where we can tell you things about quantity of DNA, about the breakdown of DNA, how different parameters would be affecting our detection possibilities. Environmental DNA, eDNA, is a non-invasive tool to understand aquatic biodiversity in hydropower impacted systems. And what I mean by a non-invasive tool, it doesn't require people to go out and actually collect an organism like many of the contemporary methods we use, like electrofishing, gill netting, seining, snorkeling, etc. eDNA also provides us with cost and time savings over these contemporary methods we use. eDNA is not a replacement for our contemporary methods, but rather it's another tool in our toolbox to be able to broaden the perspective of what we're studying and understanding what we're studying. We use eDNA in the hydropower space because it allows us to ask questions that regulatory agencies require, such as biodiversity assessments, to understand if a new hydropower plant is being proposed, how might that hydropower plant impact the species that are there. And so using eDNA, you can determine the biodiversity that's there that might be impacted, or hydropower plants that are under licensing procedures. And so this is our sampling tool that we use both out in the field and in our laboratory mesocosm experiments that allow us to sample the water using this tripod arena here. And we just stick it into the water and pump it up. It's a one-way pump, so there's no contamination. And it collects our DNA for us in these self-preserving filters. And then we can pop them off and put them back into a bag and bring them back into the lab and process them. And this will tell us everything that is living in this tank or out in a field site where we sample.
Hi, I'm Natalie Griffiths. I'm an aquatic ecologist and biogeochemist at Oak Ridge National Lab. My research focuses on understanding how water quality is impacted by human activities and energy technologies. And so as a kid, I was really interested in water. I grew up in Toronto, Canada, which is on the shores of Lake Ontario. And as a kid, we couldn't swim in the water because of pollution issues. And so I was always wondering, well, why can't we do that and how can we improve water quality? And so my research at Oak Ridge National Lab is really focused on understanding those water quality impacts, is also proving how we can measure water quality in the field. Water quality can mean a lot of different things. So one thing we can measure in the water is temperature. We can also measure dissolved oxygen. So this is a measure of the oxygen concentration in water. And low values mean that organisms that live in water, things like insects and fish, can be impacted negatively because of those low oxygen concentrations. We can also look at novel pollutants in water. So those are things like personal care products or pharmaceuticals, so caffeine, ibuprofen. We're starting to learn how those compounds affect water in the organisms that live in the water. Other novel stressors are things like plastics and microplastics. And here at Oak Ridge, I'm really focused on water quality in terms of nutrient pollution. So when we look at nutrients, we're primarily talking about nitrogen and phosphorus. And so in low concentrations, nitrogen and phosphorus can have beneficial impacts on the environment. And so the base of the food web, things like algae and bacteria, they take up those nutrients and grow, and that feeds the rest of the food web. But in high concentrations, these nutrients can have negative impacts. So one well-known example is the nutrient issues that are in the Mississippi River watershed in the United States. And so if we look at the Mississippi River, it comprises about half of the country. It's draining about half of the country, and a lot of that land is in agricultural land, things like crops, corn, soybean production. And so when these crops are grown on land, fertilizers are added to the crops to have them grow. But if you add those fertilizers in high levels or in excess, the plants are going to take all those nutrients up. Those nutrients can run off the fields into streams that are nearby those agricultural fields. And eventually those streams go down to the Mississippi River and eventually to the Gulf of Mexico. So once those nutrients end up in the Gulf of Mexico, they can cause algae to bloom. You get these large events of algal production. And then those algae eventually die and start sinking down in the water column where they're colonized by bacteria. The bacteria then start eating up the algae and respiring, which means they take out oxygen from the water column. And so that lowers the oxygen levels, which is not good for the fish, the other organisms that live in the coast. And so this actually causes what we call the dead zone each year, and we get these really low oxygen concentrations that can impact the fish and the fisheries in the area. And so this dead zone is not just specific to the Gulf Coast, but also coasts all across the world have these dead zone issues. And so we're really interested in understanding how those nutrients end up in the streams that then drain to these larger rivers into the coast, how those nutrients cycle in the environment, and then also how they're transported downstream. And so we can measure those nutrients in different ways. One easy way that we can do that is to go out and grab a water sample, bring it back to the lab, and analyze it for nutrient concentrations. But it is a very time and labor intensive practice. So if I wanted to look at a stream and understand its nutrient dynamics every day for an entire year, I'd have to go out to the stream, grab a water sample, bring it back to the lab, analyze it, do that again every day. And so one of the new advances in water quality monitoring is sensors. We have sensors now that can measure a lot of different water quality parameters, things like temperature, dissolved oxygen, pH, and now in the last couple of years, nutrients like nitrate, which is the nutrient pollutant that is causing those issues in the Gulf of Mexico. We can put these sensors out now into the stream. They can measure these water quality parameters every minute, even every second, so we can capture these changes over short and longer periods of time. But one of the challenges of using that type of approach is that the sensor is in one location in a stream and is measuring at that one spot. But we know that water quality can change along rivers and streams. So one example might be, what if we put that sensor down right before a big pipe was coming in and putting a lot of nutrients into the stream? We're going to miss that. And so a technology we developed at Oak Ridge National Lab is actually putting sensors on a drone to be able to drive these drones up and down the streams and create maps of water quality, trying to find out where there are hotspots or issues in terms of water quality. So this is what we call the Aquabot. The Aquabot is a pontoon-style boat, and it has sensors that we've installed on in the middle that hang down in the water column. And then we can drive this boat up and down the stream with a remote control, and we can start mapping those water quality parameters. There's a GPS unit on the boat as well, and so it gives us the latitude, longitude of where we are, and we can pinpoint then the water quality measurements made by the sensors to those specific locations. So all the data are connected to this data logger in this housing right here, and then we have a light sensor in the front also to look at the light dynamics. And the cool part about it too is that the data logger, where all the data then go from the sensors, has a Wi-Fi signal. So we can walk along the stream and look at our phone and look at the data in real time as we're driving up and down the stream. And so it can be used in streams pretty much as wide as it is. We need about a foot of water, and then anything deeper, it responds pretty well to that. So we developed this Aquabot system to understand some of those water quality issues in the agricultural Midwest, and specifically look at how bioenergy as well as conservation practices might improve water quality in this area. And so one of the things about the water quality issue in the Midwest is that a lot of the area in the past was wetlands. Those wetlands are drained and then converted to agriculture, but there's still an issue of flooding. And so if there are flooding events, this can damage the crops that are grown, and that can negatively impact the farmers. There's active drainage of these agricultural lands by things called tile drains. So what these are is there are pipes that are put underneath the ground, under the soil, and so water percolates down through the soil, gets into these pipes, and those pipes carry water directly to a stream. So if you're walking up an agricultural stream, you might see these pipes coming in side by side as you walk up, and so those are bringing water and potentially any excess fertilizers that are applied on the fields and then get down into the stream. And so one conservation practice that farmers and conservationists and land managers are looking at is what's called a saturated buffer. And so these tile drains still come on the fields, they drain the water and any excess nutrients, but instead of going directly into the stream, they enter another pipe that runs alongside the stream in a grass buffer strip. So grasses that are planted between where the stream is and where the crops are planted. And so now this water can go into that buffer strip, potentially fertilize those grasses, which could be grown for bioenergy. And so now you have a win-win situation where you can grow potentially another crop, but also reduce those nutrient inputs that go directly to the stream. And so we use the Aquabot system to evaluate how those conservation practices improved water quality. So we drove the Aquabot up and down the stream before those buffers were put in and also after to see if there are any changes. And so that's just one of the many projects that we're studying here at Oak Ridge National Lab focused on water quality. Sensor technologies continue to improve and our science moves forward. I'm really excited to see what future findings we have in terms of how humans and energy technologies influence water quality.